Hello, my dear students. Welcome back to another learning lessons in my Physio-Pulse channels. Today we'll discuss about the enzyme kinetics in a simple, easy to understand way as well as the exam-oriented way. You'll find the clear explanations, diagram, important concepts here. Before we begin, if you find this video helpful, please like, share, subscribe to support the channel. Now let's begin our enzyme kinetics. Enzyme kinetics is the study of the chemical reactions that are catalyzed by enzymes. In the enzyme kinetics, the reaction rate is measured and how gates changes in response to changes the experimental parameters such as the substrate concentrations and enzyme concentration. These are all the factors. The oldest approach to understanding about the enzyme mechanism and remain the most important. So this concept was earlier concept, but the latest concept, that is the if we design the initial velocity V0 and S is the substrate concentrations, then we'll get the Michaelis-Menten equation. So here S is much greater than the concentration of enzyme and can be measured MM kinetics. So how MM equation can be derived, that I will explain here. So see, first of all, enzyme first bind with the substrate, it will form the ES complex, then ES it will dissociate to form the enzyme plus product. K1, K minus 1, K2, these are the constant. V0 is determined, V0 is the initial velocity. If it is, then V0 can be calculated by this equation. V0 is equal to d by dt into ES, that means the P product, and that is called the K2 plus ES. So if we have to calculate, if we have to find the value of ES, then we will get the velocity of the enzymes. And K1 is the rate of formation of ES, K1, and K minus 1 and K plus 2 is the rate of breakdown of ES. So in a steady state, the rate of formation and rate of dissociation both are the same. So we also highlight here the equation number 3. This is the rate of formation and this is the rate of dissociation. So if you have to equalize both these equations, then we will get the value of Km. Because in one side there is the K minus 1, K plus 2 by K plus 1, that is called the Km. And this E and S, ES and P symbolize the enzyme substrate and enzyme substrate complex and the product respectively. Now this Km is called the Michaelis-Menten constant. It is a substrate concentration at which V0 is the Vmax, half Vmax. That is, half of the active sites of the enzyme are filled. Different enzymes have the different Km values. They are typically ranges from 10 to the power minus 1 to 10 to the power minus 7. The unit of Km is the concentration of the Km affected by the temperature, pH, ionic strength, and the nature of substrate. So if the V0 is equal to half Vmax, then Km is equal to S. So Km is measured the substrate affinity of the enzyme. Now let us see how this Km can help to derive the Michaelis-Menten equation. This is the Km value, and this Km value, putting this E into the equation number 5. E value is this is 0 minus ES. So this you can put here, and then we will get finally ES is equal to E0 into S divided by Km plus substrate S. So then putting the value of ES from the equation number 5 and equation number 2, the rate of V0 can be expressed in terms of S. So we will get V0 is equal to K plus 2 into E0 initial enzyme concentration and substrate concentration divided by Km plus S. And Vmax is equal to K2 into E0. So finally we will get V0 suman Vmax into substrate S divided by Km plus substrate S. The equation number 8, it is called the Michaelis-Menten equation for a single substrate or only one substrate enzyme catalyzed reaction. So in this way we will get the equation of Michaelis-Menten equation. And if we plot this V against the V0 against the substrate concentration, then we will get these Michaelis equations graphical representations. What will we get? That is S. When S is a substrate concentration affinity is increasing, then the velocity also increasing. But certain to some extent, when substrate concentration, if we have to increase, then the maximum velocity is maximized. So there the saturation has come. So this is called the Vmax. So this equation describes the kinetic behavior of enzymes that exhibit the hyperbolic dependence of V0 and S. So it is called the Michaelis-Menten kinetic. And this equation particularly determines the value of the Km and Vmax and also describe the analysis of inhibitor actions. But double reciprocal plot is more convenient procedure to determine the approximate value of Km. That we will discuss in our next lectures. So So students, if you found this lecture helpful, do not forget to like, share and subscribe to the channel. Press the notification bell so you never miss our upcoming physiology, biology as well as biochemistry lectures. And I think you all have learned or understood about the MM equation and the graphical representation of MM equations. And thank you for watching and we'll see you in our next class. Thank you.